All right, wonderful. Well, thanks a lot for the opportunity to speak today. And today I'm going to talk about our desire here, our common desire to sense microbial activity in the soil and in the rhizosphere. And we would like this to be real time, in situ, non-disruptive, and also at relevant scales to the processes we're studying. And so here I'll present a flexible along with collaborators at Aerodyne Research, volatile compounds and their isotopes as indicators of biological interactions and nutrient transformations. And so what we're trying to address here is a gap in measurement scale of our looking at microbial activity in soils. And we've heard some really fantastic talks looking at all different scales using really creative methods. When it comes to using volatile indicators of microbial activity, so these are trace gases that are cycled by microbes as metabolites or for communication, we have a gap in our measurement scale around the sort of low centimeter scale, where we have flux chambers that can resolve tens of centimeters, and some very selective electrodes that have a limited set of volatile metabolites that can be detected. And so what we're working with are some soil gas probes that we connect to above ground trace gas analyzers. And we install them at soil at different depths or locations or treatments, for example, proximity to rhizosphere, to resolve differences in these volatile indicators. And this is an overview of how this approach works. For a number of years, folks have used diffusive membranes such as this that allow gas exchange between the soil environment into this inner chamber within the membrane. And that inner chamber can then be conducted using a clean air gas stream out of the probe and into a gas analyzer that resides above ground. And so this is one example of a flow-through design where you would sample one gas probe at a time and get an equilibrated sample, and each of the steps that was measured by the trace gas analyzers would represent a different sample. And so what really we're trying to do here is to use this really massive suite of trace gas analyzers that have been developed largely for atmospheric chemistry techniques that are very precise for measuring small molecules and their isotope logs or volatile organic compounds, and integrate them with these small gas probes. And so the advances that we've made have really been to try to address the mismatch and some of the challenges that come with this integration. So for example, laser spectrometers also often have relatively high sample volume demand with their large sample cells. So in some of these reports I've listed below, we've done things like create a 3D printed volume insert that fills up the space that's not needed by the laser path. A lot of work on just achieving really controlled flow and different types of sample transfer to the analyzer, trapping that sample for observation. And we're really interested in preventing advection across that probe, which could suck gases out of the soil or add a lot of gas in, which is not our objective. And so we've done a lot to control that flow, prevent advection, and really build this system that allows us to make non-disruptive measurements. And the soil probes, there are a lot of different materials that we can choose from. We've been using sintered PTFE, which is a fairly robust material that can go into the soil system. It's hydrophobic, and working on different methods for installing these. And so we have demonstrated the use of the soil gas probing method first in some controlled soil columns. I'm showing the results here. And looking at the nitrogen cycle in particular, because it's a complex cycle that I'm sometimes still wrapping my head around, but there are a number of volatile indicators of different intermediates within the nitrogen cycle, such as nitrous oxide, nitric oxide, and NO2, that we can track. And what we find with these soil gas probes is that we're able in the subsurface to resolve really some interesting dynamics that happen, for example, here in response to wetting and nitrogen, from this below ground perspective. And we also integrated isotope analyzers for nitrous oxide with this method. And so what we found is that subsurface, we have these great pulses of nitrous oxide in response to wetting and nitrogen addition. And we can also watch the shifts in isotopic characteristics of this nitrous oxide over time, including the preference for the heavy nitrogen to be on the inner versus the outer nitrogen position. And that site preference is really exciting, because using that measurement in coordination with these other isotopes, we can actually map these different isotopes into this 3D map here, where we overlay different characteristic for known nitrous oxide production pathways. And so in this way, over time, we can look in situ and use these characteristic signatures to build hypotheses about which microbial processes are driving these nitrogen transformations in soil. And this is something that we've first deployed in the field at Biosphere 2 during a large drought experiment that we ran a couple years ago. And in response to re-wet, we found we were studying the sugar palm rhizosphere that has a lot of great exudates being pumped down to the microbial community versus an area outside this rhizosphere. And by placing the probes really directly in these contrasting regimes, we're able to see differences in the nitrous oxide emissions that occurred after the re-wetting, and shifts in the site preference, which are indicating a shift from some of the microbial nitrous oxide production mechanisms to potentially more role of the abiotic hemodenitrification. And so this technique has been often to apply, and recently we turned our focus to agroecosystems, which are really important as the primary source of anthropogenic nitrous oxide. And in these systems, this loss of nitrogen as nitrous oxide costs money, and it also releases a potent greenhouse gas to the atmosphere. And so improving the nitrogen use efficiency of nitrogen is really essential to making biofuel crops viable economically and from a global change perspective. And so in this system, we are really interested in the different plants that are used for biofuel crops. And we're recognizing that the nitrogen losses are really driven by the plant microbe interactions, not the microbes alone. And that there are different plant genotypes that are being developed for biofuels and other agricultural practices that differ above ground. They can have very different productivity, as shown here in the plant height. And we anticipate that those differences extend below ground to the microbiome. And some preliminary data collected by our colleague Betsy Arnold has shown that indeed for sorghum at this site in central Arizona, we do find that there are shifts in the relative diversity of bacteria to fungi and to endophytes between these high and low productivity sorghum lines. And so I think what's really interesting in agroecosystems is that there's this really growing, I guess, exploding capabilities in terms of phenotyping these plants and evaluating different genotypes, different sorghum lines, and their interactions with the environment and how they produce different phenotypes, which are important for biofuel production. And that's something that's done here at the Maricopa Agricultural Center, referencing platform, which actually moves across this gantry. And in that box, there are a lot of different sensors that are remotely characterizing the plants and the different genotypes and their phenotypic expressions. And so, for example, this gantry can look from the plant to field scale at differences in fluorescence, 3D plant structure, and derive results such as this, where parts of the field that are well watered versus water limited have very different photosynthetic characteristics here determined by the chlorophyll fluorescence. But while there are these above ground phenotyping approaches, the below ground approaches to really look at characteristics below ground, including nitrogen transformations, have been lacking. And so we were hoping to bring this system to Maricopa and ask if we could resolve differences in the below ground nitrogen cycling of these different phenotypes of sorghum, comparing 3 different productivity levels to an unplanted plot. And we hypothesized that as the more productive sorghum lines would have a greater nitrogen demand or greater nitrogen competitiveness. And so actually there would be less nitrogen loss in the gas phase from those lines. And that loss would increase as the productivity of the plants diminished in plots that didn't have plants at all. So we looked at 3 different heights of plants or 3 different lines, and this is showing the differences in the productivity between these replicated plots where we were studying. And what we did is we installed the soil gas probes directly beneath where the plants would grow at 10 centimeters and also at 30 to 35 centimeters in these different plant treatments. And I think one important characteristics of using soil gas probes is that we can place them within the rhizosphere to sense the transformations, whereas with a flux chamber, it would be really difficult to really resolve those transformations from the surface and even to get close enough to the plants in these dense agricultural sites to make that measurement. And so we installed these soil gas probes in the bare field before the planting began. This was a huge effort in a very hot and dusty site. And the soil probes were installed at 2 different depths. And then we installed an instrument shed that housed a nitrous oxide and NO isotope analyzer and also a VOC analyzer nearby to our plots. So I wanted to show some of our emerging results from the study. We're actually still in the field. We just harvested last week and have a couple more weeks of observation. But over the course of this cropping cycle, we have sort of 3 different stages that we've observed. The first stage, we were applying irrigation and nitrogen fertilizer to the plants through the fertigation drip line. And so that first phase was really characterized by nitrogen availability from exogenous inputs. The second phase, we have a season in Arizona where we got 21 centimeters of rain in that couple months, followed by a dry season. And so this is what we're seeing with the subsurface probes. We first see in this first phase a really rapid loss of the applied nitrogen, that those pulses are really tied to the timing events of the fertigation or additional irrigation events. And then in the second phase during the monsoon, we see this persistent loss of nitrogen as nitrous oxide, even though there's no new fertilizer input. So this is residual nitrogen in the system that is being lost under waterlogged conditions that then transition to a dry season where we see continued loss from the unplanted plots and not from the planted plots any longer. And this is just to zoom in to show the types of dynamics that we're resolving with the probes. And we can contrast this with our observations of nitric oxide, which is really tied to the application of fertilizer. We get a lot of losses of this important photochemical chemical. And but then actually during the monsoon and when there's no new fertilizer applied, we really see that NO crash out. And so seeing very different dynamics for different gases. And so what our initial question was whether or not we would see differences in the below ground nitrogen phenotypes in terms of, for example, the magnitude of nitrogen loss. And these are early results, but we've just taken the average of the nitrous oxide concentrations that the soil probe saw. And we do see this nice relationship that as plant productivity increases, we see lower losses of nitrogen as nitrous oxide. And we especially see this in the shallow depths at that 10 centimeter depth. It was really, I'm trying to show in the plot or the picture on the right, is really in the center of this dense rhizosphere of the sorghum, whereas the slightly deeper probe, maybe we see similar trends, but it's a little bit hard to tell. And so this is exciting to us that we do see in terms of nitrogen recycling a relationship with plant productivity. One other angle that we took is to look at the isotopes of the nitrous oxide and nitrogen gases in the rhizosphere, because we're interested in the difference in the magnitude of that nitrogen loss, but also understanding if there's differences in pathways and microbial processes that are interactively with plants causing this. So we did track the natural abundance of the nitrous oxide isotopes, and this is something that the data analysis is ongoing. But I wanted to show some under 2 events. We actually did a 15 nitrogen isotope labeling where we added the ammonium nitrate, which is our fertilizer, but with either of the nitrogen atoms labeled or an unlabeled control. And we added that directly into the rhizosphere and tracked how that was converted into the gas phase in situ, and are also interested in how that nitrogen is assimilated into plants and microbial biomass and their products. And so this is a picture of after the monsoon season, after nitrogen hadn't been applied for a couple of months, we added this labeled nitrogen precursor, and we see these nice dynamics of conversion into the nitrous oxide of that label. In this case, we saw that there was the most label from the ammonium precursor, and that actually the high productivity line had the greatest conversion. Before the monsoon, we actually saw some very different dynamics where we had, for example, an earlier peak of the 15N label, a lower overall label, and that actually the low productivity lines had more conversion. And so we're still really digging into these results, but we think that this is a promising method to look at the rhizosphere in situ and see how nitrous oxide is moving throughout the nitrogen cycle. So to summarize, the below ground phenotyping of the plant of the rhizosphere using the soil gas probing is able to capture dynamics in nitrogen cycling at really nice spatial scales, and also that those are resolved by depth and plant genotype in ways that I think make sense and are exciting. And that we do find that in terms of our science questions, that the nitrogen losses and the pathways seem to be differing with sorghum genotype. And we're really interested in continuing to ask what the detailed roles are of the plants and the microbes in these interactions and in these overall results. And it's probably more than just plant nitrogen demand. How are these organisms interacting? And so we're really thrilled to have recently been awarded ficus support through EMSL and JGI to continue to ask these questions, and looking overall in the shifts in the roots and the microbiomes. Can we see how these nitrogen transformations and activity are differing with genotypes and cropping stage from other angles? And that includes some genomics, metabolomics, and imaging. And following the 15 nitrogen approaches, can we look at which of the pathways of the nitrogen loss are more significant, and really where is this loss or these conversions localized in the rhizosphere between the roots and the microbiome? And here we hope to try 15N DNA SIP metagenomics. We know that this is going to be really challenging, so have plenty of other great analyses planned. But yeah, really looking forward to having all these great capabilities to build a more complete picture of the system. So with that, I'll just thank the team and a lot of other folks who have helped us out with a really quite grueling field campaign, and wanted to acknowledge our funding. This has been primarily supported through a DOE SBIR grant led by Aerodyne, and I've also had support through the University of Arizona. Yeah, and I think I'll go ahead and stop there if there are any questions.
Laura, thank you for a great and invigorating talk. There is a question here. Could nitrogen fixation in the rhizosphere be measured using these soil gas probes?
Yeah, we think so. We haven't tried that at all. For example, with like 15 and labeled N2, or maybe even conversion to acetylene, right? But yeah, I think these open the door to a lot of in situ observation and run the probes, kind of in reverse, and diffuse, dose out or diffuse out your precursors of interest. So we think there are a lot of opportunities to use them both for sensing and for dosing your precursors.